Okay. Well, that'll work. That'll do. It's fine. Thank you for attending my talk today. We're actually at the tail end of collecting a lot of different behaviors. So I'm really excited to give an update on kind of where we're at right now. Perfect. Okay. So though we're in an opioid epidemic, there's still an increasing use of psychostimulant and cocaine use. And it's not just with people that are using multiple substances. So it's not just through the combination of psychostimulants plus synthetic opioids. It is also cocaine alone. And currently there's no accepted treatment for psychostimulant use disorders, but we do know that there's some evidence to suggest that they're highly heritable, but we don't know much about the etiology beyond that, mainly because there's kind of a lack of power and sample size in human GWAS studies right now. But luckily, since we work in rats, we have the ability to identify new genetic links to psychostimulant use disorders in the rodents. So in this project, we capitalize, kind of like everyone else, in the session has been talking about, on the fact that inbred strains themselves also have substrains within them that can be very similar genetically, but still express different phenotypes from each other. And these are magnitudes genetically closer than, an inbred strain would be to another inbred strain. And this allows us to map genetic differences to the behaviors that we're interested in much faster than we would in other circumstances. And this is thought mainly due to occur from, genetic drift over being in different facilities for over a couple decades. And so the two substrains that we're looking at are from the spontaneously hypertensive rats. So we're using the substrains from Charles River and Harlan. These are not the stroke prone ones. These are a different subset of the SHRs. But we found that one of them, the Charles River SHR, show more of a pro-addiction phenotype on a variety of different tests. And so they're showing these high levels of impulsivity and compulsivity, but they're also self-administering drugs of abuse, a variety of different types of drugs of abuse at a much higher rate. And we don't see this with the SHRs from Harlan. The majority of the time, though, these projects have been looking at SHRs compared to inbred strains or outbred stocks, so they haven't been directly comparing the two together. However, thanks to some funding from the University of Tennessee, we were able to have a project where we did start to compare them directly to each other. So this was a previous paper that we had looking at male SHRs from both facilities, where we did find that the Charles River SHRs had more of this pro-addiction phenotype and self-administered more cocaine compared to the Harlan SHRs. So building off of these results, in order to map the genetic differences between the two that relate to the behaviors that we've identified, we produced reduced complexity crosses. That is, we start out with our parental substrains, which are very genetically similar, but again, different. And they are phenotypically different, which we've shown already. And then we cross them together twice to create our F2 generation, to shuffle the genome, which allows us to determine if the presence or absence of a specific loci relates to some of the results that we see in our behavioral battery. So not only do we run males and females from both of these generations, but we're also tracking whether the initial cross to create the F1s was from a female from the Harlan substrain and a male from the Charles River substrain, or vice versa. And the reason why we're doing this is because we're maintaining these lines separately to determine if there's any multi-generational parent of origin effects in the F2 generation. And so the first step in our timeline is to breed the rats in-house to get rid of any environmental variance between the two vendors and to produce our parental and our F2 generations. So then we take them through a variety of assays that probe for potential differences in things like reward preference or novelty responses and impulsivity. This timeline starts with our cocaine locomotor assay. And in this assay, the animals are given first injected with saline, and then it's followed by 2 doses of cocaine, a high and a low dose, over the course of 2 weeks. So on the first day of locomotor, they're injected with the saline. We found that the parental substrains do differ from each other in that in the first 5 minutes that they're in there for a total of an hour. The first 5 minutes, the Charles River SHRs are locomoting much more in response to their environment compared to the Heartland animals. This is also something that we saw previously when we were getting them directly from the vendors, that the Charles River had more of this novelty response. And the reason why we care is, novelty responses are one of those things that are often comorbid with addiction, even if they're not directly related to taking the drug. Further, when looking at sex differences and adding on the F2 generation, we find that there's both a main effect of group and a main effect of sex, in that the females are locomoting overall more than the males are. But the F2s seem to be showing and within the sexes, the substrains are significantly different from each other. And then our F2s are showing a kind of an intermediate phenotype in between the two. So they're not significantly different from either substrain, but they are showing kind of an in-between. So a different pattern emerges, though, when the rats start actually getting the cocaine. And so in the first week, they get a high dose of cocaine. And in this week, all rats locomote more over time. So on days 2 and 3 compared to day 1, they're all sensitizing to the repeated doses of the cocaine. We're also seeing that overall, the females locomote more than the males in both generations, as marked with all of these pound signs. That's relative to the females. But instead of the F2s in this case showing an intermediate response between the two parental subtypes, there seems to be a sex-specific difference with the F2s, in that the males seem to be, and a lot of these aren't significant, but the males are kind of locomoting less than the two substrains, but the females are having the opposite effect, in that the females are actually locomoting more than the two parental substrains. And it does become significantly higher than the Harlan substrain by the end of the week. And the Harlan substrain is the one that we expect to be lower addiction phenotype. And so this pattern of behavior continues into the second week when they're getting the 5 mg dose. And actually on this dose, every single day, the females are, the F2 females are significantly higher than the Harlan females. So that's like I said, the Harlans are the substrain we predict to be on the lower pro-addiction phenotype. But what's interesting is that when we look at sucrose preference data, we have the opposite kind of reaction, in that the Harlan animals are showing a greater preference than the Charles River animals to sucrose. All of the animals choose to drink more of the sucrose than the water, but their preference scores are the thing that's higher. And then you'll notice that this time, however, I split up the F2 generation, and that's because doing the complexity crosses in the way that I indicated, like I said, we're tracking which lineage they're coming from. And so the F2s that come from the Harlan sires are showing significantly higher sucrose preference than the F2s coming from the Charles River sires, which I found particularly interesting because, although not necessarily significant, if you look at the 4 different parental groups, the highest ones are the Harlan males. So it looks like the highest ones in the parents are the Harlan males, and it's their offspring 2 generations later that are showing the highest amount of sucrose preference in the F2 generation. So due to a bottleneck on resources, only the parentals have made it this far in the timeline. So we do a couple other assays, one of which is an operant assay, in which the animals are lever pressing in an operant box to get a chocolate pellet reward. This is a differential reinforcement of low rate responses in behavior, DRL. And so they have to inhibit their responding for 5 to 30 seconds. And the idea is that if you're much more impulsive, it's going to be harder to inhibit your responding for that amount of time. And we do see that in the parental animals. The Charles River animals have a lower response efficiency, meaning that less of their responses are actually being rewarded. So more of them are just restarting the timer. And then additionally, we are looking at burst responding. So not only can they not wait 5 seconds to press, but burst responding is less than 2 seconds. So they are really rapidly pressing when they're trying to seek the pellets. When it gets to the DRL30, however, There's not significant differences, but as you notice, all of them are doing really poorly. They can't wait the 30 seconds. It's a harder task for them. So we don't see any substrain differences anymore. And then this is the most preliminary of the data. The ends are very small. It's actually really half the animals that we ran through self-admin because we were running yoked saline at the same time. So all of these had an additional animal that was yoked to them. But what we're seeing currently is that, after we do a surgery to implant a catheter in the femoral vein, which runs subcutaneously underneath the skin to a head mount, the animals are able to lever press on an FR1 schedule for a moderate dose of cocaine. This is just an FR run for 14 days, and this is at the end of their 14 days. And we are seeing some evidence to suggest that there's a difference between the substrains as well here, in that the Charles River animals are likely going to earn more infusions and also press the active lever more to get the cocaine, which also does fit with our previous paper, which was just in the males. So, in conclusion, the Charles River animals are showing these higher novelty responses and impulsivity, and likely will end up self-administering more cocaine than the Harlans, whereas the Harlans are showing a greater preference score for the sucrose preference task. The F2s depend, they're showing an intermediate novelty response compared to their parental substrains, but they're showing this kind of female-dependent increase in acute cocaine sensitivity relative to either parental substrain. And then there's also evidence of a multi-generational parent of origin effect on sucrose preference in the F2s. So we actually run even more behaviors. So we run a lot of other behaviors. It's a pretty comprehensive timeline. We're going to, in the future, be doing a couple different other schedules on the intravenous self-admin and doing a cocaine response curve, as well as other drugs of abuse in these animals. And then seeing if we can predict the IVSA from the premorbid, the novelty, those types of behaviors. And then our next step essentially is going to be putting a pause on behavior. We've collected a ton. And so we have, about 250 plus animals that we've done all these dissections on that we need to start doing the genotyping and getting into the actual genetics underlying everything. And then, so I want to thank everyone on the slides. Like Cameron said, I'm actually currently still at BU. I'm still a terrier, but I will soon to be a husky with him. Everyone that has an asterisk next to them was directly involved in collecting the behavioral data. Emma gets 2 because she has done a lot. She's one of our techs. And then again, thank you for the funding. Some of it was from Tennessee, the others from NIDA, and also 2 of our undergrads during the summer got some funding from their individual schools at, well, one of them was from NIDA, and then one of them was from Kenyon College. So with that, I'll take any questions.